Alright, so chapter 9, we're going to go over physical and chemical means of controlling microbial organisms. Alright, so in terms of either controlling their growth or outright eliminating microorganisms. Alright, so we're going to begin talking about some terminology. Alright, so one of the first things is sterilization. Alright, when you're sterilizing a substance, alright, either it be culture media or glassware or surgical equipment, what you're trying to do is you're trying to kill and remove all microbial organisms. Now this also will involve endospores. Alright, so when you're trying to sterilize something, you're trying to remove both endospores and bacterial cells. Alright, the vegetative bacterial cells. Alright, so you can do this through a process of using a autoclave. An autoclave is nothing more than a homemade, basically, pressure cooker. Alright, it's the same concept. High temperature, high pressure. Alright, it's the high heat that's really doing most of the destructive work. In this case, the high heat denatures proteins, which ultimately kills a cell. Now, pressure can also do the same thing. It's also going to disrupt cell structures, particularly proteins. Alright, so disinfection. Alright, so this could be either a physical process or a chemical process that eliminates vegetative cells, but it will not eliminate endospores. Alright, so using a disinfectant to disinfect certain types of tools used for a job. So, you know, a hairstylist typically has their scissors and combs and other tools and some type of barbicide. Alright, disinfect it. Alright, so whenever we're in the lab, alright, and we spray down our benchtop with a dilute concentration of a disinfectant or bleach, alright, we are trying to remove vegetative bacterial cells, alright, but we will not kill if there are any endospores present on the benchtop. Now, typically, you apply disinfectants to inanimate objects or materials, alright, not to living tissues. Alright, so decontamination or sanitization is basically cleaning. Alright, you're trying to reduce the number of bacterial cells to a safe level. Alright, so for example, when you wash your hands, alright, even when you wash your hands, alright, you don't eliminate all the bacterial cells that are on the surface of your skin. Alright, there are some that are still left there. Alright, but you're trying to reduce the number of bacterial cells to a safe level. Alright, so you can decontaminate, alright, using certain types of solutions, equipment used in food processing plants, eating utensils. Alright, so think about a restaurant. Alright, you typically sanitize or decontaminate, clean all of your equipment and all of your utensils, all the glassware, all the forks, all the spoons, all the pots and pans. Everything gets clean or sanitized. Alright, washing your hands can be also an example of sanitization. Alright, so long as you're using soap or detergent, alright, even a alcohol sanitizer solution. Antiseptics. Alright, antiseptics are used on living tissue. Alright, so here you're trying to outright kill or inhibit the growth of microbes. Alright, so flushing out a wound with, for example, this Band-Aid solution of antiseptic wash, or rinsing your mouth with Listerine are examples of using an antiseptic. Alright, betadine or iodine is another example of an antiseptic, particularly that's used on wounds in a hospital or doctor's office. Alright, so the use of iodine compounds, alright, there's more than one type than what you pick up at your grocery store. Alright, there's different derivatives. To repair the skin for surgery is known as which of the following? Disinfection, antisepsis, sterilization, sanitization, or degermination. Well, here, iodine used on the skin, antiseptic. Alright, so antisepsis. Alright, so different types of microbial organisms inherently on their own are resistant to different types of antimicrobial agents. Alright, so you notice I'm using the word antimicrobial agents. Alright, so that's a very broad term, alright, because it's all encompassing. Alright, so your most resistant antimicrobial agents are things like prions, alright, which aren't even living organisms. Alright, they're proteins. They're abnormal proteins. Alright, this requires the application of chemicals and extreme heat to eliminate the presence of prions. Alright, so this has actually been a pretty big topic in the past couple of years. Alright, because there have been numerous hospitals that have accidentally potentially infected patients with the use of surgical tools that were not adequately cleaned, sterilized, all right, because they were used on a patient, and unbeknown to the physicians, they didn't know that patient suffered from a prion-associated disease. All right, and later on, those same tools were used on another patient, and so you have the potential of crossover. All right, spread and contamination to other patients. Alright, so your endospores are the second most resistant specimens. Alright, so in this case, you can eliminate endospores using extreme heat and pressure. Alright, so autoclaving, alright, usually does the job. Alright, in fact, when you talk about autoclaving, one of the ways you can tell whether or not your autoclave is doing the job right and sterilizing everything is you use control specimens with endospores. And if those endospores are dead at the end of an autoclaving process, then you know that your autoclave is working right. Alright, so it's kind of a way of standardizing and looking for quality assurance and a quality check that your autoclave is up to par, that everything's working as it should. Alright, your mycobacteria. Your mycobacteria, in this case, have, if you remember, mycolic acid, alright, in their cell wall. And this mycolic acid is going to be highly resistant to all different types of chemicals. Alright, so it's very waxy. Alright, so it's a lot of lipids. Staphylococcus and Pseudomonas are pretty hardy pathogens. Alright, they're pretty difficult to eliminate. Alright, then you have your cyst and trophozoites of your protozoans. Alright, your gram-negative bacteria. And then you have your fungal specimens. Alright, so when you're talking about these guys here, your fungi, alright, fungal spores. So think about it. If you're using some type of disinfectant, cleaning agent, stuff like that, keep in mind that you're dealing with a eukaryotic specimen. Alright, so you have to be cautious in what you're using because you don't want any type of potential high toxicity to us, essentially. You have your non-enveloped viruses, your gram-positive bacteria, and your enveloped viruses. Alright, your enveloped viruses are the least resistant. Alright, so recall that your gram-negative bacteria have this outer membrane of basically lipids, right? Alright, things called LPS, lipopolysaccharides. And so this outer envelope structure of the outer membrane makes this group of organisms more resistant than gram-positive bacteria to disinfectants. They're a little bit harder. Alright, so this table, alright, the same table as in your book, it gives you a nice overview of all the different types of control measures. Alright, you can use physical means of eliminating and controlling microbial growth. You can use chemical agents, and you can use mechanical methods. Alright, and we're going to walk through these different types of processes in the next couple of slides. Alright, so two big terms that you need to come to grips with are bacteriostatic and bactericidal. Alright, so bacteriostatic agents. Bacteriostatic agents only are going to inhibit the growth of cells. Alright, so notice here that when you add your bacteriostatic agent, your bacterial cells stop growing. Alright, so whether you're looking at a total cell count or you're looking at a viable cell count, alright, it flatlines. Alright, so bacteriostatic agents inhibit growth. They don't necessarily kill the organism in question, but they slow down their growth. Alright, your bactericidal, alright, so think about like this end part, side. Alright, it's something like homicide, you're killing. Alright, so here, a bactericide kills the bacterial cells. Alright, so here, if you add your bactericidal agent, notice that your total cell count remains the same, but your viable cell count, your viable living cells, drops precipitously. Alright, so you're killing bacterial cells. Alright, so what is microbial death? Alright, how do we define death in a living organism? Well, for humans, nowadays, alright, you can check for a pulse, alright, you can check for a heart rate, heartbeat, and you also check for brain activity, alright, but here we're dealing with microbial organisms, so how do we know when they're dead? Well, a microbial organism is considered dead when it can no longer reproduce at optimal growth conditions. Alright, so it can no longer undergo binary fission. Alright, so what type of factors would affect the death rate of a microbe? Well, this is something we'll get into in terms of looking at different methods of eliminating pathogens. Well, one is going to be the load, the number of bacterial cells you're dealing with. All right, whether you have a small number of bacterial cells or you have a high number of bacterial cells. It's also going to depend on the type of microorganism you're dealing with. So if we're talking about, you know, a virus or a gram-positive bacterium compared to something like a endospore or a prion, one of those groups of organisms is going to be more difficult to eliminate than the other. All right, so the type of organism is going to affect the death rate. All right, the type of method you're using. All right, so whether you're using a physical, chemical, or mechanical method to eliminate microorganisms. All right, because you have to use a certain type to eliminate a certain group or type of microorganism the best. And then you also have to deal with any other type of inanimate objects you're dealing with. Alright, so where are you trying to eliminate the microorganisms? Is it on a living tissue in a living organism? Or is it on some type of inanimate surface, like toys or dolls or stuffed animals or a bench top? The type of surface you're dealing with, the type of substance you're dealing with, is going to affect what type of antimicrobial measure you use. Alright, so there are different cellular targets, right, that these physical and chemical measures are acting against. Alright, so specifically looking at a bacterial cell, alright, you have particular types of agents that can target the cell wall. Alright, so think about what makes a bacterial cell unique, or what makes it stand out differently from a eukaryotic cell. Alright, well, here, the cell wall. You can inhibit cell wall synthesis. Alright, you can inhibit protein synthesis in bacterial cells. You can disrupt the cell membrane of a bacterial cell. You can disrupt the bacterial cell's metabolic pathways. You can disrupt DNA replication and transcription. And then you can also prevent the pathogen from either adhering to or recognizing a host. Alright, so these are all different cellular targets that physical and chemical control agents are targeting. Alright, so your textbook walks you through the different types of germicidal agents. Alright, I'm going to go over a couple of these examples specifically. Alright, but just kind of familiarize yourself with things like, for instance, bleach, which is a halogen. Alright, so sodium hypochlorite. Alright, what does it do? Well, sodium hypochlorite is basically one of which it's going to be an alkaline. It's a base. Alright, so sodium hypochlorite can denature enzymes. Alright, that's one of the big things. Alright, so you can use it to kill bacterial cells, endospores, fungi, viruses. Alright, there's different methods. You can use gaseous chlorine or liquid chlorine. Alright, so hydrogen peroxide. Alright, so hydrogen peroxide, keep in mind this is a reactive oxygen species. Alright, so it's a liquid. Alright, you can purchase at the department store. Alright, so in this case it can target DNA, it can target lipids, it can target proteins, and it can disrupt those structures. Alright, so it's toxic to the bacterial cell. Alright, alcohol. Alcohols target the cell membrane. Alright, so lipids are soluble in alcohol. Alright, so in this case, you can disrupt the cell membrane of the bacterial cell. So you effectively disrupt that barrier between the outside environment of the cell and the inside environment of the cell. Alright, so looking at surfactants. Alright, so surfactants are things like soaps, for instance. Alright, so a soap is able to intercalate into the membrane. And when it intercalates into the membrane, alright, because your surfactant has a polar end that's usually got a charge, and it's got a non-polar end. Alright, so it intercalates into the membrane. And so basically it creates this pore in the membrane. It makes the membrane more porous. It makes it more leaky, so that substances can enter and leave the cell. Alright, basically it disrupts that barrier. Alright, so looking at physical control measures. Alright, so one of these would involve heat. Alright, so there's two different types of heat. You have moist heat and dry heat. Alright, so moist heat involves use of hot water, boiling water, or steam. Alright, dry heat, you're talking about an open flame or candle. Alright, so when you're talking about use of temperature, there's some terminology you'll want to come to terms with. Alright, one of which is the decimal reduction time. You have the thermal death time. Alright, so the thermal death time is basically the time it takes to kill all of your microbial agents, microbial cells, at a specific temperature. Alright, so the shortest period of time it takes to kill all the cells at one temperature. You have something called the thermal death point. Alright, so thermal death point is basically the lowest temperature in which you can kill all of your bacterial cells within 10 minutes. Now, something you might come across more often is the decimal reduction time. The decimal reduction time is the time it takes to reduce the population by 90% at a given temperature. Alright, so here you're basically looking at a 10-fold decrease. Alright, so since we're looking at a logarithmic scale, so we're going from 100 to 10, the decimal reduction time against some arbitrary organism at 50 degrees is about 40 minutes. At 60 degrees is around 15, and at 70 degrees is around 8 or 9 minutes. Alright, so by this point in time, you have eliminated 90% of the viable cells in your culture. Alright, so if you start off with 100 cells, well, you're left with 10. Alright, so basically 0.1%. Alright, so the thermal death time for this would probably be around 10. This one around 40. And then 50 is going to be a little bit more on out there. Alright, so the decimal reduction time is commonly used in food microbiology. That's one of the many applications of it. Alright, so you have two organisms, alright, A and B. One of these guys is a little bit more hardy and resistant to particular temperatures than the other. Alright, so organism B is a little bit more resistant to the temperatures that are used than organism A, in terms of reducing the population size by 90%. Alright, so here you're also looking at decimal reduction time. Alright, so these are what we call D values. Alright. So here you're looking at number of microorganisms per gram, minutes of cook time at a particular temperature, at a specific temperature. So here 140 degrees Fahrenheit All right, decimal reduction time going by tenfold reduction is about 2.5 minutes. All right. In terms of completely eliminating organisms, you're looking at around 8.5, 9, 10 minutes. All right. But at higher temperatures, the decimal reduction time, right, because from 1,000 to 100, it's the number of organisms per gram. That's basically a 90% decrease in the number of organisms. All right, it's around a minute or less. All right, so looking at the effect that heat has on microorganisms. All right, so we've already talked about proteins, and we've already talked about the effect that different types of environmental factors can have on protein structure. All right, so here we're talking about denaturing proteins. All right, so you can basically take this tertiary or quaternary protein structure, denature it into its primary structure. Or you can alter its shape or disrupt its ability to interact with other substances, and it's no longer active. So basically what you're doing is you're causing these proteins to denature and coagulate. Alright, so it's basically essentially like frying an egg, is the idea.
This familiar gelatin dessert actually is a good example of the process of coagulation of proteins into a three-dimensional latticework that entraps water molecules to produce a semi-solid gel. This animation explains the physical chemistry behind the process. Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are three structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. The molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein. The assembly of irreversibly denatured protein molecules results in formation of a solid gel. The gel entraps water molecules inside the white into a semi-solid structure, which holds its shape under normal conditions. Other examples of denatured protein assembling into three-dimensional structures include the baking of yeast-risen bread, coagulation of meat proteins by cooking in such products as hot dogs, and in the solidification of gelatin upon cooling of a solution. Denaturation and coagulation of proteins is a complex, irreversible process, but the study of denaturation has allowed us to better understand the three-dimensional structure of native proteins.
All right, so looking at process of autoclaving. All right, so here, all right, you have your samples that you're autoclaving, whether it be glassware or it be waste materials or other tools you're using. Right. Basically, you have this sealed container that's under a vacuum. All right, so you have an outer jacket and an inner jacket. And initially, you have this outer jacket that's filled with gas, steam, and what happens is once you shut the door and let it start, you set your parameters. Right, so typically 120 degrees C, 121 degrees C, 15 psi. Typically for 15 to 30 minutes for autoclaving. You have this steam that comes in along with heat. You have this vacuum that pulls out all the air, so it creates this vacuum. So high temperature, high pressure for a long duration of time.
The term sterile means free of germs and disease agents. Sterilization refers to the process that removes living microorganisms from materials and objects. The goal of product sterilization is to kill all microorganisms and spores. In addition, viruses, prions, infectious proteins, plasmids, and other DNA fragments are destroyed. This makes a sterilized product non-infectious, meaning you cannot catch diseases from it. Steam sterilization. Heating in an autoclave is standard procedure in most hospitals and doctors' offices. You see the sterilizer being loaded with surgery containers holding sterile goods. The containers serve as storage until the next patient treatment. The materials to be sterilized are treated with overpressure for a certain period of time and at a certain temperature. For prion destruction, the materials that are being sterilized should be heated to 134 degrees Celsius for 18 minutes at 3 bars of pressure. The air in the autoclave interior should be completely replaced with saturated water vapor. These sterility conditions are only guaranteed with Category B sterilizers. These create a vacuum inside the sterilizing unit before the sterilization process begins. This means that air is sucked out and replaced by water vapor. This guarantees that the materials being sterilized are surrounded by water vapor on all sides, an important condition for sterilization. Ask your doctor if he uses a Class B sterilizer, as these are, unfortunately, not the norm. If the doctor does not have a Class B sterilizer, it is recommended that you abstain from undergoing treatment because the instruments are not sterile. After sterilization, the containers are removed from the autoclave, and the surgical instruments are prepared for upcoming patient treatment. In the picture, you see a surgery container in which the instruments are neatly stored. Doctors' offices do not usually use surgery containers. This leads to an interruption in the sterility chain as soon as the instruments are removed from the autoclave and come in contact with hands and fingers. Sometimes the instruments are shrink-wrapped in a special paper foil that allows for short-term sterile storage of the instruments. However, the package cannot be damaged or the instrument immediately becomes non-sterile. Sterile instruments are useless if non-sterile covers are used. There are different options in terms of sterile covers. The use of disposable articles is safe, as these are sterilized by producers through radiation and are thus free of germs. Prior to surgery, you see how the instruments are sorted on a sterile disposable cover that serves as padding. Frequently, washable linen cloths are used instead of disposable articles. These are sometimes sterilized in an autoclave, but some doctors' offices do not do this. Either way, the linens are not sterile because even an autoclave cannot completely remove germs from the linens. How do you find out if you're in good hands with your dentist in terms of hygiene? Ask your doctor about hygiene guidelines. A conscientious doctor will happily answer your questions, while a dentist that has gaps in his system will not be so eager to volunteer information. Ask for sterilization protocols, Class B sterilizers, storage containers, disposable covers, and thermodisinfection device. There will be more about that in the video by the same name.
Alright, so in terms of autoclaving, what you're being shown is the pressure in blue and the temperature. Alright, so initially everything's at 0. Alright, you preset your autoclave for a specific temperature that you want to have the internal chamber reach, as well as a particular pressure. Now, as I've mentioned, the temperature setting is around 120, 121 degrees Celsius, and the pressure is somewhere around 15 psi. Okay. Now, this is a standard value. All right. These numbers, all right, the temperature and the pressure can be altered. All right. It's going to depend on what you're trying to sterilize. All right, the particular contents, what those contents are in, the organisms you're dealing with, all that's going to play a role on the temperature used and the pressure, as well as the amount of time. All right, so usually we spend around 15-30 minutes. So this duration actually lasts for about 30 minutes in time in terms of a cycle. All right, so usually when you're talking about sterilizing glassware or culture media that you're going to use in a lab, it's usually around 15-30 minutes. All right, that's normal. That's typical. Now, if you're going to autoclave to sterilize glassware, or not glassware, but waste, right? So trash, all right? So bacterial cultures or old culture plates, things of that nature, they have to be autoclaved for a longer period of time, usually up to for at least an hour. Okay, so as the temperature of the chamber is increasing, all right, the pressure also begins to increase. You have pressure that you have basically steam that enters inside the jacket of the chamber as the temperature increases. And then you have a period at which you have sterilization. All right, so a period of 15, 20, 30 minutes. All right, once the sterilization time is reached, all right, your jacket, the internal jacket of the autoclave begins to release steam gradually. It's a slow process. All right, and as it releases that steam, the autoclave also begins to cool off. All right, and once it cools off to around 100 degrees Celsius and your PSI reaches 0 PSI, you can then remove the contents. Now, keep in mind, your contents are still boiling, so they're very, very hot. So they have to be handled with care. Alright, so coming back to this idea of endospores. Alright, so your endospores are the second most resistant cellular or microbial structures. Alright, so you have basically the inside, the core, basically the genetic material to that little bacterial cell inside of it. Alright, the little bitty bacterium. Alright, little bitty bacterial cell that's encysted in the spore. And you have these outer layers of cortex, alright, which basically contains nutrients for that little developing bacterial cell, calcium. Now, your endospores, they are resistant to heat. Well, they're highly resistant to heat. They're resistant to radiation. All right, so what happens? These endospores, you actually have water that's been removed out of the core of the cell. All right, so there's very little water activity. All right, so here you're looking at the application of dry heat to moist heat. All right, so dry heat, it takes a higher temperature. All right, so here if we kind of standardize what we're looking at, so comparing moist heat to dry heat. All right, so here you're looking at, let's say here, level bacterial cells. You're looking at a longer period of time at a higher temperature to eliminate endospores with application at dry heat than moist heat. It takes a lower temperature. To reduce your level of endospores by 90%. All right, notice here that your endospores also persist for a longer period of time. All right, so coming back to this idea of how we know that an autoclave is functioning as it should. All right, so here you basically have this little ampule with little bitty endospores in it. All right. And what you do is you crush the ampule after autoclaving, and basically the ampule contains nutrient broth. And if you see turbidity after incubating this little capsule for a day or two, that means your autoclave isn't functioning as it should, because the endospores basically released the little bacterial cells, the little vegetative cell, and it began replicating. Now, if you killed the endospores, then the culture media remains clear. All right, and that's how you know that your autoclave is functioning like it should. Now, pasteurization. All right, pasteurization, you're basically going to be using, there's different types. All right, there's different durations. So you can take like milk, typically pasteurized, and you heat it, all right, to kill most of the microbial organisms. All right, so you're basically using this indirect heat. So you use a period of hot water that's going to transfer heat to the milk. You heat up the milk to around 72 degrees from refrigerated temperatures, and then you begin this cooling process by giving cold water that basically absorbs the heat from the milk to cool it off back to refrigerated temperatures. All right, so if something is pasteurized, that does not mean that it's sterile. All right, so pasteurized milk, pasteurized juice, all right, these products are not sterile. All right, if you let them sit in the fridge, all right, they spoil. All right, not to mention you're dealing with organic materials, they decompose anyway. All right, but you have microbial organisms that begin to use that milk or use that juice as a nutrient resource, and they begin to populate. All right, so you can also use freezing, all right, to kill microbial organisms. All right, so you can use a process of quickly freezing products versus slowly freezing them. All right, so if you quickly freeze a product, all right, you have very few ice crystals that form. All right, which is great if you're talking about something like a food product that you want to keep intact, like ice cream. Fewer ice crystals, all right, smoother it is. All right, but in this case, the cell wall of your whatever you're freezing remains intact. All right, a slower process of freezing, you have larger ice crystals that form. Those ice crystals damage the cells, they kill the cells. All right, so typically products are frozen pretty fast, all right, to maintain the integrity of the cells. So think about food products you buy at the grocery store, or products that you freeze yourself in a Ziploc bag. All right, think about if it's been sitting in the freezer for a long period of time, you have these large ice crystals that form. And if you use that particular food product for a meal, the taste is a little off. Right? You might have freezer burn, but then also those cells that make up whatever that product is, if it's ground beef or it's vegetables, it isn't going to taste right because cells have been damaged. All right, but most of your microbial organisms, all right, are highly susceptible to freezing conditions. There are your psychrophilic and psychotrophic organisms that can, either replicate at freezing temperatures or survive freezing temperatures. Alright, so things like Listeria. Alright, so here you're looking at a graph of mesophilic and psychotrophic bacteria during cold storage at 4 degrees Celsius on poultry. Alright, so notice here your mesophilic bacteria, alright, they have this really long lag phase. Alright, for 10 days at 4 degrees, they haven't grown very much. After 10 days, they increase a little bit, but not much. Your psychotrophic bacteria, after 5 days, begin replicating exponentially. Alright, so this is why it's a good idea to follow the instructions on the product labels of foods. Alright, so you're told, for instance, like hot dogs. Alright, you're told that after so many days, you need to have consumed those hot dogs after opening that package. Well, the issue is things like Listeria that can contaminate your ready-to-eat food products like hot dogs will again to at refrigerated temperatures multiply. Alright, because what you have done is you've taken that vacuum sealed package that has no oxygen and you've opened it up and you exposed it to the air. So you've basically introduced oxygen into that package. And so now the organisms begin replicating like crazy if they're able to multiply at 4 degrees. Alright, so you can also use a process of lyophilization. Alright, so freeze drying. Alright, so talking about frozen foods. Alright, so you can take your food products and they're flash frozen. Alright, so you basically draw out almost 98% of the moisture from the food product at really, really low temperatures. So you do this so quickly that you maintain the integrity of that food product. Alright, so it's basically like you're picking that green out of a field, or you're taking that cutlet straight from the cow to the plate. Alright, in this case it's flash frozen. Now you also have this process of lyophilization. And this process of lyophilization, where you're basically freeze drying. And a lot of your bacterial cultures, alright, that you get for a lab nowadays are freeze-dried cultures. Alright, so what you do is you basically have pulled all the moisture out of those cells, and you've done this at a really low temperature. And what we do is we take those lyophilized cultures and we rehydrate them and incubate them at the appropriate optimum conditions to reculture them. Alright, so looking at radiation. Alright, so a lot of your food products nowadays are going to have this little symbol showing you that your food product has been irradiated. whether it be ultraviolet light or gamma rays. All right. Each of the particular types of radiation have their own respective uses. All right, so in terms of using something like UV light, that's only so good on a surface. It's not very penetrating in terms of going into a product or cells directly. They don't penetrate very far. All right, your gamma rays are usually what are used for food products. They penetrate much deeper. All right, so here you actually have a bacterial culture of Serratia, and what happened is you have this little like cutout, all right, that you place over the top of the culture. You irradiate the culture with ultraviolet light, and then you place it into the incubator. And basically, anywhere that was exposed to the ultraviolet light, you see there's very few bacterial cells. All right, because they were killed. All right, so we'll talk about what ultraviolet light and radiation does to a cell. All right, but here looking at strawberries. All right, so 25 days after treatment at storage 30 degrees Celsius, so refrigerated storage. All right, so looking at the levels of radiation, which are measured in grays, in this case, we're talking about kilograys. All right, so the control sample that was not irradiated after 3 days, after 25 days at 3 degrees Celsius, so almost a month, you have fungal growth. All right, they're completely spoiled. 1 gray or 1 kilogray, all right, they're starting to spoil. All right, 1.5 kilogray, they look good. All right, you have a sample that was heated for 10 minutes as control. It's showing spoilage. Heated at 10 minutes plus irradiation at 1 kilogray. It looks perfectly fresh. All right, so sometimes you may see that different types of physical and chemical methods of treating products is effective. You may have this synergistic effect where you can basically reduce costs for using a process by combining things together like heat and irradiation to meet the perfect efficient method of eliminating your pathogens to increase the shelf life of your food product. Alright, so what you're being shown here is the different types of microorganisms that are sensitive to radiation. Alright, so looking at the number of the D10 values. Alright, so D10 is telling you, alright, basically the amount of radiation that's needed to reduce initial population or the activity of the organism by tenfold. Alright, so your D10 value for Clostridium botulinum, Clostridium tetani, alright, Bacillus. Alright, these guys are capable of forming endospores. Alright, so that's why they have such a high level of radiation that's required to eliminate them. Alright, so it takes this high of a value to eliminate reduce the population tenfold. So from 100% to 10% survival. Alright, organisms like E. coli and Salmonella, which are gram-negative. Alright, you have gram-positive organisms like Lactobacillus. You have Aspergillus, which is fungus, a mold, Saccharomyces, viruses, enzymes. Alright, some of these require much higher levels of radiation. Alright, notice here your Deinococcus radiodurans. This organism is almost comparable to that of Clostridium in terms of the level of resistance to irradiation. Alright, so what does irradiation do? Alright, well, irradiation, such as UV light, damages DNA, for one thing. In this case, you can have a thymine dimer. Alright, so here, you have a normal segment of DNA. Alright, so you have two strands of DNA that are complementary base pairing. Everything's normal. Is exposed to UV light, now you have your two thymines on one strand of DNA that are now interlocked. And so when the DNA replication machinery comes in to replicate the DNA, when it encounters this thymine dimer, the replication fork stalled. Alright, so basically now you have this covalent bond that's linking these two thymines. Alright, so this is where your DNA repair mechanisms come in. Things like, for instance, photolyase. Alright, this is a light-dependent enzyme. Comes in and basically removes that covalent linkage. Alright, you also have a light-independent process called nucleotide excision repair, where the cell will come in and remove some of the nucleotides and then replace them with the appropriate nucleotides. You also have a process of mechanical control of microbial organisms, so filtration. Alright, so in terms of filtration, you're taking a liquid and you're filtering it through a porous filter. Alright, so you're going to use something like vacuum filtration. Now, in this case, you're able to filter your samples based on the pore size of the filter. Alright, so you can filter out cysts, filter out bacterial cells, but in this case, even your viruses can pass through your filters. Alright, so you have to use some other type of physical or chemical control method to eliminate your viruses. Alright, because they're so small, they can pass right through your filter. Alright, you can also use a syringe to filter solutions. Now, in terms of filtering a solution compared to actually using a type of physical or chemical treatment, well, think about it. A solution that is highly susceptible to heat or radiation or some type of chemical treatment, if you were to alter the properties of that solution in some way or inactivate it, alright, that's not going to be very functional. Alright, so sometimes you have to actually filter solutions. Alright, that's the preferred method of sterilization. Alright, because you want to maintain the activity of your respective solution. Alright, a lot of times you'll filter sugar solutions or filter antibiotics. Even sometimes vaccines, you can use some type of filtration mechanism. Now you can also use salt or other solutes. Alright, you can alter the osmotic pressure. So you increase the osmotic pressure of a solution by either adding solutes that are soluble in that solution, like salts or sugars or things of that nature. And you basically increase the osmotic pressure on the cell, basically drawing out moisture from the cell. Right? So this is the reason why we have foods, specifically foods that have a long shelf life, that are preserved, that are usually have high salt contents. Even solutions or even food products that are frozen or have high salt contents. All right, so that's something to keep in mind when you're buying those frozen foods, is they have a high salt content as well. All right, this is there to reduce the level of activity of microbes. All right, in terms of their ability to replicate or even survive. All right, so different organisms can survive in different amounts of water activity. All right, so things like fresh meat, milk, all right, bacteria can survive in. All right, but then talking about yeast and molds, all right, they can survive in jams and jellies or fruit concentrates. Some bacteria, like your halophilic bacteria, all right, in this case, you have more solutes or lower water activity. All right, so here, things like honey, flour, dried fruits that are dehydrated. So think about it. As you dehydrate something, what are you doing? You're basically concentrating the solute in that food product. So that food product has a high concentration of solute. All right, so that's why dehydrated foods have a pretty long shelf life and keep well. All right, so this idea applies to canned foods like Spam, all right, as well as canned fruits, vegetables, jams, jellies, preserves. In this case, what you're doing is you're drawing all this moisture out of the cell. So you're causing the cell membrane to pull away from the cell wall. We call this process plasmolysis. All right, so which of the following physical control methods would you use to sterilize the following items? All right, so in terms of inoculating loops, all right, inoculating loops, you're going to use a kind of dry heat. All right, we use a flame, right? So here we have dry heat. All right, for the inoculating loops, for glassware contaminated with bacteria, you're going to usually use, a lot of times, a disinfectant, and you're going to autoclave. Okay, vaccines, all right, you can use filtration. You can also use, in this case, radiation, all right, for vaccines. And then powders and oils, you're going to use hot air oven. All right, so in this case, you're going to be using a form of dry heat. All right, so chemical agents. All right, so your chemical agents occur in a liquid, gaseous, or solid state. All right, so it's important to keep in mind that your chemical agents, that they can act as disinfectants, they can act as antiseptics, they can act as sterilants and preservatives. All right, it depends upon the concentration of a respective chemical solution and what you're applying it onto as to what type of antimicrobial agent you describe it as. All right, now the question becomes, how exactly do you choose the right microbial agent? What are things you got to keep in mind? How do you know which one to select? Well, what are you trying to apply that chemical agent to? Right? What are you trying to treat? Are you trying to treat an inanimate object? Are you trying to treat living tissue? Are you trying to treat glassware? Is it something that is perishable? Or is it something that you're going to reuse? If it's something that's perishable, well, you're going to want to use probably a method that's going to kill everything that's contaminated that object, it'll destroy it, but it's going to be trashed anyway, so it's not an issue. But if it's something you reuse, you're going to have to choose the right or the appropriate chemical reagent that isn't going to destroy or damage or somehow alter the properties of whatever you're applying this chemical agent to. So you don't want it to be corrosive. All right, you want something that's broad spectrum. All right, you want something that is going to have very low toxicity to humans. You want to make sure that whatever you're using is going to remain active in the presence of organic materials. All right, because the presence of organic materials lowers the activity of certain solutions. Also, you got to keep in mind the nature of those chemical solutions themselves. What is their shelf life? How active are they? All right, something like bleach. When you make up bleach, mixing water and bleach, the activity of that solution is good for maybe a day. After that, it's no longer active. All right, so you have to make a fresh batch. You got to keep in mind cost. You got to keep in mind the availability. What are you using it on? How much of it do you use? Is it used on a regular basis, or is it something that's used very rarely? You have to keep in mind its solubility. Is it soluble in water? Is it soluble in alcohol? What type of shelf life does it have? How well does it penetrate inanimate surfaces? Alright, so looking at chlorine gas specifically. Alright, so it's soluble in water. Alright, so you could get something like hypochlorous acid. Alright, functions as a disinfectant. Alright, it can basically pass through the cell membrane and disrupt the metabolic cycle of the cell. Alright, so the fact that it's an acid, it can disrupt the internal pH of the cell. You can end up with other variants of chlorine, so hypochlorite, ozone, which is an oxidizing agent. Alright, so it can damage lipids, damage proteins, damage DNA. You have hydrogen peroxide, another oxidizing agent, another reactive oxygen species. Hydrogen peroxide, so in this case here, you have these reactive oxygen species that are natural byproducts of metabolism. Alright, so in this case, you can damage proteins. Alright, proteins that are involved in metabolic pathways. If you disrupt the proteins, you shut down the metabolic cycle of the cell. The metabolic cycle of the cell is shut down, well, the cell dies. It can't synthesize the things it needs, like amino acids. You can also have, as you're damaging these proteins, you're releasing iron, because you do have some proteins that have iron centers. These iron centers, the iron interacts with hydrogen peroxide to form hydroxyl radicals. Hydroxyl radicals are the most damaging reactive oxygen species. Alright, in this case, this hydroxyl radical can also damage DNA, which is lethal to the cell. Now, cells do have a way to basically disrupt or reverse this process of reactive oxygen species. They have things like superoxide, dismutase, which will take your oxygen radical and converts it to hydrogen peroxide. Alright, You then have an enzyme called catalase that can convert the hydrogen peroxide to basically water and oxygen gas. Alright, but in this case, your hydrogen peroxide can damage DNA. There are DNA repair mechanisms. In this case, release of iron. So your oxygen radical can convert it to hydrogen peroxide. You can have the formation of hydroxyl radicals that damage the DNA as well. The cell has to repair the DNA damage. It also has to repair damage to proteins. Silver nitrate. Alright, so looking at metals. Alright, so we know that certain metals have antimicrobial properties. Alright, so silver nitrate is usually administered to infants, right, whenever they're born. Alright, so in this case, you can have, as the infant passes through the vaginal canal, if by chance the mother has a venereal disease like gonorrhea, all right, she could pass on the gonorrhea bacterium, the Neisseria gonorrhea bacterium, to her infant. And one of the first places they would come into contact is the eyes. All right, so this is one route of exposure. In this case, if it infects the eyes, you end up with this infection in the eyes, so neonatorium ophthalmium. All right, so in this case, you have this basically conjunctivitis that's due to the bacterium. And so one way of preventing this is by the administration of silver nitrate to the eyes. So here you have coins that have silver in them. And so here you can notice that there is a zone of inhibition around the coin. So the bacterial growth has been stopped where that coin is located, and the silver in the coin has actually leached out to the media, inhibiting the growth of the bacteria. Alright, so which of the following antimicrobial agents are considered sterilizing agents? Alright, well, in this case, looking at chlorhexidine. Alright, chlorhexidine is mainly used as a antiseptic. Alright, hydrogen peroxide. Alright, it is usually an antiseptic, alright, which is basically the 3% concentration. Alright, but you can get some concentrations that are around like 35%. This is going to be more commercial. In this case, this would be a sterilant at that concentration. Alright, so notice that at 3% it's an antiseptic, but at 35% it's a sterilant. Alright, so alcohol is mainly a determinant. Alright, so usually in the form of a sanitizer. Alright, acetic acid. You can use acetic acid in this case like vinegar. Vinegar is going to inhibit the growth of most microbial organisms, so you use it typically in like canning. Alright, so you're not going to necessarily eliminate everything,